Okay, I think it's right day. We are ready. Okay, today we are going to talk about three topics, separated topics: batch effects correction and pseudotime analysis, and also repertoire profiling analysis. So the batch effects problem it's not a new problem for single cell data. So in the traditional RNA sequencing, bulk RNA-seq, people realize this could be a problem, so they want to use the batch effect correction. But in this case, the example I showed is from this publication 2018, and they have a control sample, which are in the orange, and this pale color cells is from this challenge F and beta. I don't know what it is, but they believe they're batch effects. So they make the correction and align the data set, make all the cells look like they all superimpose onto each other. I believe in this process they changed the expression levels of all the cells to make them match towards the same aligned profile somehow. Some of this batch effect correction is only based on the embedding. So they change the position of the cells, make them superimpose onto each other, and then you do the clustering. So you put same cell type, but from two different batches into the same cluster. So there's two different levels of those. It's called harmony algorithm. But in general, so this is a blog publication, and the authors showed the data points before and after the correction. He used this called a combat. This is a very old algorithm that's even before single cell data become widely available. So combat is for traditional bulk RNA-seq sample, and these two, based on the nearest neighbor cell, the neutral nearest neighbor, and the CCA, which method introduced in Surin package, also try to achieve the same goal. So we will mention this method. But that's the quotation I just quote. Just be careful, okay? I never use it. I just do individually cell type identification. Then they merge by the same cell type, but they're still in the same different conditions. Unless you believe there should be batch effects. What I tell you what I did before, I just convinced myself there's no technical batch effects at all. What I did is in our lab in early 2018. We did LCL studies, LCLs. Those are the lymphoblast cell lines. We get a sample from Europe and the sample, and then Africa. Two cell lines. Send this to a single cell RNA. So this is my sample one. I did this for sample two. I also put 50% of this, 50% of this, mixed. My sample three. So I did this. Of course, this is on the same machine and the same batch. There's no difference. So in data analysis, you can separate African cell line from European cells very clearly, and you can group them. So their difference, and no batch effect difference. Indistinguishable. You mix them and sequence separately based on their genetic background and their profile of the expression. Okay. I think unless you believe there are very strong reason you believe, okay, this machine or you send a sample to this center to sequence, and all the samples, even though you gave this kind of mixture, or you know they will control, they come back and may not emerge. Then you believe there's strong technical bias, and you probably in that situation you want to do some kind of batch effects correction. And for the done appear and their samples from the cancer, and if you believe, okay, this is a healthy individual, then you have two controls from individual samples, and you try to push them together, that will create completely distorted data sets. And then use this to compare, for example, the sample from patients and tissue from different type of different stage of the cancer. I don't know, but that's just don't do it. Okay, but anyway, so batch effects occurs when non-biological factors in the experiment can change, okay, to change the data produced by the experiment. So that's the reason. So that's mentioned the combat and MNN, surrogate, CCA, and some other algorithms. Okay. And in this blog, and the author say, okay, sometimes this very simple old-fashioned batch correction algorithm works really good. Okay, so you don't have to introduce new algorithms for single cell. Just borrow the old ones. It's also okay. But if you want to use more fancy approach, this is published 2018. So it's called mutual nearest neighbor. Let's see how it works. So we have two batches of data, and one is in this faded out color, and you have 3 clusters. And another is batch 2 here. Okay, this algorithm will look at nearest neighbor. So based on their embedding, okay, you can use 3D or even high 4D, 5D, depending on how many features you want to keep. But anyway, you can compute the distance for all the cells from one cell to all others, and have a pairwise. And then you have a label, you know those cells are from batch 2 and those from batch 1. So you found the cells in batch 2 and link to the nearest neighbor in batch 1. You found the link. Okay, so in this case, in this subpanel B is try to find those links. Okay, the cells in this right cluster have a nearest neighbor in another batch is in this group, so you make a link. The mutual nearest neighbor is you go the other way around, you found the cells in the batch 1 and look at the nearest neighbor in batch 2. If they're the same cells, then you keep that link. Otherwise, you just, if it's just one direction, you don't keep it. So after you do this, and also you know how to compute the distance between cells. Okay, there's a norm between one vector against another, so very easy to compute. Then you just keep those records, and then you make those alignment, basically a link between cells belongs to different batches. So once you finish this, you put the cells, then move into the same space. Okay, you cross them down. And then so this is to look at the neutral nearest neighbor. So once you finish this, once you have batch 3, you start to do that again. Okay. So in this case, you finish the batch effects correction. Okay, you just push their mutually nearest neighbor links and push the cell to the same space. You move them. So this is very easy. It's very intuitively straightforward. So another technique people use is called canonical correlation analysis, so CCA. And you can see the results here, and then you basically you do a PCA for batch 1 and do a PCA for batch 2. But this PCA is not consider individual batch data itself, because if you only consider batch 1 data, you will rotate the data to show the maximized variability. Well, in batch 2, you do this independently, then you show the maximized variability, but that's not the case. We try to rotate the 2 data points at the same time and found their respective rotation matrix to allow them to align as much as possible. So sometimes it's not the most variability you can see after dimension reduction, after rotation is the best. You have to consider 2 batches at the same time, let them to rotate to an angle, then you see them maximize align, some kind of correlation. So that's why correlation is in the middle. So you want to maximize the correlation between two batch of data points after their respective rotation. Okay, so in panel B here, it shows this: do the CCA first, rotate your cells, allow them to align, and then because of this scaling effects, you see this all the cells in batch 1 seems to shrink a little bit. So you want to realign them. They use a technology called dynamic time warping. So DTW, we will mention this after the CCA introduction. So you try to rescale the stretch around the data to make them align. And then you recompute their position to make them the cell aligned and put in the same clusters. So we are going to look at some technical details in the CCA. And there are more generalized application for CCA. I think as a biologist, you're better to have this idea. So CCA is not just rotate, align 2 different batches of data. In this case, this is cell batch 1, cell batch 2. They are same nature by the data of generation. But you can use the CCA for more generalized concept. So as long as you have this semantic objects, okay? Let's give you some examples. So you have a view for the data from one domain, and then you have another view for the data, a different set of data, another view. You can align them, okay? So this is like a concept picture, and then you have a completed domain of the knowledge of a view of your data points. And on the other side, you have another completely separate domain of knowledge. You can align them based on the structure of data points distribution in each of their respective domains. The example is like this. On your left is a view of a picture, okay? And on your right side is a sound wave, some kind of signal. They are completely different domain of the signal message, but it's possible to use the CCA to align those signals. Another example is on your left is a view of pictures. Okay, you have pixels, colors, shapes of objects. Well, on your right side are the view of the information. It's the word description of what's in the picture. If you can summarize all the signal or the sample data points and put it in this space of view, okay? And you do the same thing for the another view. There's a way you can align them. So this signal is rely on the difference between these two samples. For example, these two samples occupied the space in this shape, and another is here. Well, this view you can project on the space in different shape, but you can align the shape. Another example is this EEG signal and fMRI, functional MRI signal. They are different sampling technique, completely different. And from the same object, then you have two different views, and there's a way you can align them. The whole idea is you transform your data from view 1, transform your data from view 2, embedding them into the same latent space and make them a correlation as high as possible. So I want you to open your mind sometimes, okay? It's not only for aligned cells, and in future study, you may have different data technology to describe your sample in different ways. So consider that if you have enough sample, enough data points. We don't know when you will need this. So this illustration to show, okay, you have variable, this is like a view 1, you have data points like this, variable site Y, define your data points in this green dots in another space. So you use CCA to find the basic X and Y to do the rotation, okay? You rotate your data points in the subspace in this view 1 space, and rotate the 2 and the reduced so data points, and try to make their correlation as high as possible. So the information we will use, including you know what it is. This is a covariance matrix, okay, to describe the data points and their variable correlations. And this is for Y. And then we compute the correlations. So this information will be used in the optimization. Basically, the CCA, we try to achieve the correlation after the rotation as much as possible, as big as possible. So this is an expression, mathematical expression. So you have a correlation function, and you use this learned projection, the rotation, basically. It's allow X to project on this A defined new coordinate system, and also do this for Y, but use a set of different correlation correspondence. Then you try to maximize the correlation between A and B after projection, okay? And with this goal, you can define this expression and define all the variables, including this covariance matrix for X itself, covariance matrix for B, so Y, and also this covariance matrix between A and B data points. So this is expression of those correlation. And this is the major steps. So you have covariance matrix computed for X and Y. And before that, they have a centralization. You have to always centralize your data. So in that space, you remove this. But anyway, so when you do the PCA, we have to centralize data and then compute the correlation, covariance matrix. The same thing here, but you just do this for X and Y, two different batches of data. And then you compute the covariance matrix and do this, it's kind of compound covariance matrix here, and then use SVD to identify those this is called the singular values is in the sigma here. There are singular vectors, which you can use that as a rotation matrix to rotate your data. Okay. So in MATLAB, we will play with the CCA function, this canonical correlation function that take X, Y, and give you the projected results of A and B. Okay, they are aligned already. So in the lineage algebra, people realize CCA and the PCA and several other of these algorithms is based on the rotation of your data points, and then for the PCA, you just found the rotation which maximizes the variability view. Will CCA have try to get a correlation between two batches, and the underline they have a same structure, like PCA is one of the special case. You only consider one group of data points, which is the covariance for the X. And then it's more generalized in another matrix B, there's identity matrix where CCA will consider both of these separately. So the underlying greater level of this framework is shared. We can also consider CCA as an optimization problem. So you don't know the rotation, you just try to get the A and the B, and then rotate your original X and Y data points. Try to minimize this difference. Okay. Depends on your data. If you have a non-linear components on your data, you probably want to consider their kernelized CCA. Deep CCA, I believe, is using some neural network to learn. In general, what we described is conventional CCA. There are other versions of for this probabilistic, maybe better for if you have missing data in your sample. Okay. So this just show kernel CCA, including this kernel function. Remember, the kernel function is compute to make to increase the dimension of your original data by, for example, production of this XY, or this to introduce extra variables, try to help you to translate this nonlinear relationship into more linear relationship and make the separation of optimization easier. So this extra page of the slide just indicates when we talk about the CCA, if you are people from an evolutionary ecology, and there are another term called the canonical correspondence analysis. It's also called CCA, but that CCA is completely different from this CCA. So Just make sure don't get confused. They're completely different analysis for different purpose. Okay, we mentioned the dynamic time warping, and it's a technique to help you to align this time series data. Basically, if you have a sample collected with a different time, but their time points are not match, and you want to align them eventually. We will play with this example in the computer lab. So this is called a TWD. If you have two samples, for example, this blue curve is from X and this orange curve from Y. They are sampled at a different frequency. You get this kind of unaligned signals. But if you use this time warping and you reshape one of them to let them to match, and you get results like this. Okay, in the CCA application I showed you, once you after the rotation of your data points, sometimes the scale of your cloud of the cell not matched. You can make the smaller cluster and more stretched out to align with the larger ones to make the final embedding. So one of the steps is this. So this algorithm involved in the computation of the distance between data points in the two time series data. So once you have this distance matrix, you start to find the paths, which allow you to see, okay, the shortest path I should go is from this corner to there, and in the middle, I have this 3 increment of the steps, which indicate there are 3 steps in blue curve should be joined and aligned to 1 data point in the right curve. After that, you achieve this match of the alignment. Okay, so we finish off this batch effects correction part. So you have one, you decide whether you need it. Another, you have old combat algorithm, sometimes achieve better results than more fancy recently developed method for single cell data. For single cell data, there are 2 major ones introduced 2018. There are mutual nearest neighbor methods. There are CCA-based methods. Okay. If you don't have a question, we will move on to pseudotime analysis. So people always are interested in the cell lineage tracing, and especially if you study this development or differentiation process. The most direct and most example is you want to study embryo development at a different time stage. If you can sample this, I have a lot of data from zebrafish, it's easy to collect. Or you want to study some dynamic process, such as its regeneration of tissues, or stem cell differentiation, when you have a clear stage, which has a function as time. So you want to trace the change. In the cancer study, you want to trace the cancer. And sometimes people sequence the DNA to find the mutation accumulation across the time. Anyway, so there are many different methods have been developed. Okay, different kind of signals you can track the change of cell population. So once the single cell become available, people start to think about to use computational technology to apply to single cell data to understand the dynamic of the cell population across the time. Even though sometimes the samples only collected from one time point, and then because internally those cells may under different stage of development. So people want to reconstruct this trajectory to learn this dynamic process. Of course, if you have samples collected from multiple time points, you know which one is early, which one is later. That will help you. So you can do this pseudo-time trajectory for them collectively, or you do this independently, and then link those cells from one time point across to another time points. The motivation is simple. We believe there are some limited number of the trajectory. Although if you allow the cell to develop their multiple ways, but because it's genetic, we call it cannibalization, and Washington has this idea about this limited number. You start with a very diverse genetic background, but the cell will just choose some of the parts to it's either to solve the problem or adapt to the environment. So there's a way you can think about it. There's a limited number of trajectories. So you try to reconstruct this, understanding the development. So the goal is to model the transition between cell identities. Sometimes you have one cell transform, differentiate in another, or you can model differentiate process, identify the gene. So after you constructed this trajectory, and you can see, okay, cross the time, and if I look at this gene expressed in these different cells, and we found, okay, one gene increase their expression, another decrease expression. So you want to find those genes associated with the time points, which defined by the pseudo-time analysis, to infer the trajectory, to learn the biological process, okay, to find the control regulatory genes. So this will only become available if you have enough cells. Okay, if you only have 5 cells, or like bulk RNA-seq, you cannot do it. Okay, so it's only take advantage of the shared number of cells that you collected from a single cell technology. So this picture just shows the cells has their development times is color coded by color coded here, but you collect the cell group at one time point, and then you want to reconstruct this, try to reflect its true development time. I don't have any cursor here, sorry about that. And then the result is you try to find the genes change along the time frame. So this is an example for embryo stem cell from human. So you have collected the cell at time 0, time 4, 6, at a different date, and then you put those cells as a group, and then found their clusters. So the clusters, then you go through this pseudo-time algorithm to linearize, basically, make an elongated cluster. Basically, a cluster is a cloud. Then if you try to use algorithm to force those cells to be aligned on the curve, then you get something like this. What they found is you can reconstruct some of this process. I think the difficult part is you have different branches. Okay, so you go this way, that way. So it's not a very simple straight line from one point to another. In the middle, you have a branch, And after branch, you may have another branch. So I just found this is very difficult. If you follow this trajectory, so you go from stem cell and then to the green, and then go this way, or you go this way or that way. So you create many different projects. And which one you should follow? So it's really you still have to know your system very well, and you go. Okay. I mean, in data science, you can create a lot of these signals and then found, because you have so many genes, you always find some genes which correlate very well with this path and that path and this path. But how at the end you can synthesize this information is still a question, but it's one of the steps you can take. So I illustrate the points like this. If you want to take the picture of this, you have to use this camera and take the picture at multiple times. But we don't have this kind of picture. We just use a population of cell to reconstruct this process. Okay. Same idea here. If you don't have a video camera capture the video, the movie of the movement of a horse, but you want to study this position or movement of a gate, you can take a picture for a lot of horses, a group, and then realign this horse, and then probably you can reconstruct the movement with time series data. Same idea if you have a t-SNE plot, but you transform this into this elongated trajectory shape, and you may get some signal out of it. But let me jump to the Twitter quotation I want to show you at the end. Yeah, I don't have that. So this is from Raj, and again, this is from yesterday. He said pseudotime is not actual time. So he realized, he probably spent some time, realized, okay, pseudotime is not actual time, and it leads to a lot of pseudo claims. I think that's also a consensus in the field. I mean, the high end of the researchers, they realize. But as a general pipeline, you still can use it and try. It makes sense sometimes. But always keep this in mind, and when you construct it, maybe it's a pseudo information, okay? Because you tweet, you somehow use data science methods to align the cells. Let's see how they align the cells to make them projection. So this is an early stage of this publication, 2017. So I completely disagree with this kind of analysis. Okay, so they have a PCA, and then you try to find some of kind of trajectory within there. But I don't know how they do it, but they just do that. And they have UMAP, and UMAP is a cloud of this, and they construct. Well, they convince themselves, but it's difficult to look at any trajectory. But there's some center of the cluster. There's maybe more cells in this, more cells in that. They probably use those marker genes and identify the center of different cell types. Then they make a link, and they believe there's. So once you have those trajectory, you can have the paths from here to here to here to here. Once you have paths, then you can always find some genes, follow that path and have to show this kind of a change of levels. But again, the levels of gene expression it's informative, but not always reflect the function of cells. Remember, I told you the highly variable gene is more important. Maybe we should do some more highly variable change of some cells. But yeah, normally they just use average gene expression in time point 1, 2, 3. I mean, you recover the sum of the change of the average as global. But as I said, the hidden variable gene, this analyze just not capture the information of increase of variability of some cells, some genes in the cell, the subset of cells. Your average sometimes may reflect this change of variability, but not always. Okay. Okay, so this is from a review paper to see, okay, what the algorithm used in the pseudotime analysis packages. Monocall, I don't know which version this is about, but slice, T-scan, waterfall, okay, different names. So they use PCA, ICA, t-SNE to do the embedding first, to do the, so the high dimension data, first you have to project them, okay, to do the embedding first, always the first step. Then depends on the different packages, the authors select, for example, MST, construction, the curve fitting, okay, so once you have this, you can fit a curve. Here you found this nodes, and then try to link with a minimum span tree. Okay, so different ways. So after that, you start to make the link between those cell population cores. Okay, so once you have this link, you start to project cells, force them to project themselves onto the link. Okay. Okay, since they all use MST, so what is the minimum span tree? So this algorithm is graph algorithm. So if you gave it this distribution of your data points, it can create a graph. And on the graph, you can follow the paths. Okay, to minimize weight span tree is a subset of edges of a connected and weighted undirected graph. And connect all the vertices together. So if you create the t-SNE, and then you found those cores, then you make a link between them. You also compute the distance between each link. After that, you can give this algorithm will find this path. This path will go through all the nodes, but the total steps, the length is minimized. Okay, you found the path, link everything, but this is the minimum distance you can travel to make the link. Another group of these algorithms based on the nearest neighbor graph. So you make a link between cells if they are neighbors. Okay, for each point, you will find the 3 nearest neighbors. Okay, make a link. And you move to another cell to find the 3 nearest neighbors, make a link. You don't make a link for these cells to other cells which too far away, cross which this is larger than the nearest 3. So you don't do this long distance links. So you just move locally and then make a network to link every cells together. So then once you have this local step networks, then you start to move from one node to another, follow this shortest path or something like that, and to move from one end to another. So once you found this path, you can create the trajectory, then move the cell onto that trajectory, then finish this Okay, as you can feel, that's the all data science manipulation of the data points based on some of this hypothesis, not hypothesis, assumption. Okay, so this assumption is all the cells are distributed along this hypothetical trajectory, not far away. They create this local network, and then you can reconstruct the trajectory by follow some of these paths along these local networks to reconstruct trajectory. And this method is all the cells are distributed along this development trajectory. Then you can recover it using minimum span tree, even though there are a lot of branches in the development. But those assumptions may be not true, okay? Probably every assumption is wrong. Okay, so this is a demonstration of nearest neighbor graph. Let's see the time. I will see whether I can play this. This allows you to select different k. So if you have a k and n, you have to decide the value of k. So if you have a k equals to 3, then you found nearest neighbor for each point. There are 3 nearest neighbors. If you increase k, you probably create a different k network. Let's see if I can not on the screen. So there, for example, in this case, I have 99 points on this space. Now, if I want to join, so you can change the k, the 2 to 3. To 4, so you form this different local networks. Okay. Makes sense? It's very easy to understand, right? For each point, you found 4 nearest neighbor points, make a link. And then you move to another 4 nearest neighbors, you make a link. At the end, you get this. At this point, for example, will not link to this because there are 5 nearest neighbors around it itself. But if you increase this, you get a dense and dense network because it considers all the nearest neighbors. Okay. So this is an example. If you use Kn when k equals 3, so you found a isolated component. This is a component, all the data points are linked to themselves without link to any others. But if you increase k to 4, okay, this small island, this local network module disappeared because when you search top 4 neighbors and they start to make a link to join to the neighbor nodes. At the end, you get something like this. We have an application in Toolbox. You can create a local KNN network like this. All the points are the cells. So I make this network, and then I found a trajectory follow from one end to another. I mean, it's some of the game to create this trajectory along this network, and then we can, on the genes, expression change with this trajectory. Okay. So this is just like a mini version of the package I described here, not here. So this one here. To create a KNN network and then make a trajectory and then assign cells which close to any time points, just assign a pseudo time for all the cells. But there are some problems if you have a cells which goes this way. I probably should have another analysis to go from this branch. Okay. So let's take a 5 minutes break. So we have mentioned the PCA, ICA, CCA. Maybe something else. So this kind of a terminologies. Maybe in the final exam, I just have a multiple option questions. CCA is this, PCA is this for different purpose. So make sure you remember and know what they are doing. Their goal is okay. For pseudo-time analysis, people also have a different other algorithms that's scattered around the different packages, and the RNA velocity can be considered as one of the approaches. In this very special analysis using spliced and unspliced mRNA signals to assign so-called velocity values for each individual cells. Next, we are looking at the most popular pseudotime analysis package called Monaco. Well, Monaco itself, its early publication in 2014, so definitely a pioneer in the whole field. However, Monaco itself is not a stable package. It changed all the time from version 1 to 3. The algorithm used internally completely flipped, changed. So make sure you know which algorithm, which version you are using. Let's see the early stage version of Monaco 1, so when they published in 2014. They start to use ICA to do the dimension reduction. Then you to build MST, so you know MST is minimum span tree. Then order the cell using MST to align cells onto MST, the parse, and label cells by type. So that's the original Monaco. In Monaco 2, they changed this, use reverse graph embedding. I don't know exactly what it is. You have to read the paper in order to really understand it. They will change, okay? They claim that Monaco 2 will produce more accurate, robust trajectory than previous version. Okay. No prior information about the gene and the characteristic this biological process, number cell fate or branch points trajectory a design for the experiment. So it sounds like it's become more data driving and unsupervised. You don't need to provide any other information like this, you have different cell types. Okay, so what is reverse graph embedding? So we have to read that again. So to learn a parsimony principal graph. The principal graph is like a principal curve that passes through the middle of the data set, but it allows to have a branch. Okay, so if you have a cloud of data, try to find a curve in the middle to go through all the data points. This is called a principal curve. Sounds like very simple. It's not easy at all. I tell you, if you have a data point in 3D like this, to humans, very straightforward. You know, okay, there's a curve. I tell you, in algorithm, especially if you have some data points like outliers, you have great impact. So this is called a principal curve. And they use principal graph, so you have not only the curve but also have a branch. You can have data points. So like here, then you have branch. Okay, so this illustration for the flow chart, the pipeline of monocode version 2. First, we have PCA or other dimension reduction algorithm. Then it have a guess initial cell trajectory on the central nodes from some centers. Then update the cell position based on the current trajectory, and then update the cell central node. Okay, see line. Okay, so this is monocode 2. This is monocode 3. Monocode 3 try to be a more generalized tool. So in general, we talk about monocode. It's a tool which for trajectory analysis. But for monocode 2, since the authors claim they can do clustering and differential expression analysis. Of course, the trajectory analysis is still the most important function in the package. The algorithm build is based on UMAP and the Lovin. This is a network community detection algorithm. So basically, they change the algorithm again. So internally, without telling you which one is good, which one is bad. To me, it's just some of kind of pipeline or games. You how to manipulate the data and in the way. So maybe misleading. So another slingshot also has been considered to be one of the best. So it also involved in the minimum span tree, principal curve, so all those technologies. So in their paper, they compare the other pseudotime methods, including model 1, model 2, and it's several other KNN based. Okay. They explain the difference between algorithm. Okay. So model 1 use the longest parse through the minimum span tree. So the others do the cell cluster first, then build the minimal span tree across clusters. Well, slingshot, you instead using this principal curve. Okay. So they gave some examples, say, okay, slingshot, they reconstruct more this more realistic development stage because they have this well annotated branches, okay, from a blood cell, I believe. T-scan is another unsupervised algorithms. Use a minimal span tree, cross dimensionality reduction, PCA. The paper was good, so you can follow the paper easily. And then I did this algorithm implementation completed in MATLAB, just follow the paper. So the function is available in the toolbox. This is another algorithm called a stream. So that's a pipeline. I'm not going to go into details. There's a link for the paper. I don't hear about this. People use this package. Lisa is another one created by one of my friends, and they never release the source code, but the principle is like this. They use this landmark isomap. We will discuss isomap on the next lecture about the manifold analysis. But anyways, all kinds of data points manipulation technique. And then you construct minimal span tree on the cluster and estimate the pseudotime. There are review paper to put all these different kinds of pseudotime inference algorithm side by side to compare their performance. They consider different kind of input, the data with a branch or not, whether they're circle or in their trajectory or not. And they consider overall score or accuracy or scalability, whether it takes forever to run some thousands of cells or not. So different metric to compare the large numbers of these packages. There are conclusions. I mean, it's gave a very complicated results like this, and I think sometimes this algorithm better than another, okay? But in general, it's complicated. I don't remember what their recommendation is, which one. But in general, people just use monocode for, okay, because most of the people accepted monocode results, but just make sure which version of monocode you're using. Monocode because it's early, so most of people use it, not ask for anything else. They do have a practical guideline for the method use. I think they have some questions whether whether you expect their circle in the topology, well, it's really. So there are another package developed in R, they're called Daniverse. Daniverse try to collect all the methods on the market to put it under a same umbrella. So you have installed this, you can run all different kinds of this algorithm under the same framework. I don't know how they did this. It's kind of a engineering. Okay, so after you reconstruct this trajectory using pseudotime analysis method, so next you can first look at this trajectory, the shape, okay, the topology. So from one cell type to another subtype and which others. Then you can visualize the relationship. And most importantly, you want to identify the genes that respond to or associated with this change along the time. Again, just remember this, you may create a lot of false claim just because of you fitting your data. Nicely use one of this method. So we also consider to use this method. So this is from my own. So we can have a trajectory of cells, for example, like this. Okay, then I'm going to sample the cells using sliding window. So this here I create this multiple sliding window along this trajectory. Cells in each window, I will consider them as one group. So for each group of cell, I will create gene regulatory network for all the genes in the cell, and then I move to another group of cell along the trajectory. In that case, I will create multiple networks. Okay, so I put this multiple networks, so this is 2D adjacent matrix, and then put them along the trajectory. So I created this a qubit of data. Okay, 2D network, but you have multiple 2D networks, so the 3D structure. Then there are tensor-based decomposition method. Okay, so this is just like SVD, but it's tensor-based. So you create three components. The multiplication of the three vectors create a qubit. Okay, cubic matrix is tensor. So just like if you have two vectors, outer product is matrix, but you add another dimension, you create the whole 3D array of data. We try to learn this, but in one of these components at a time. So we can either use that time framework or not to reconstruct this for downstream analysis. Okay. Some applications, illustration like this, we can do this cell type specific of this time series network analysis. This is a neuron, so the trajectory I created, maybe not a time, but maybe this different layers of brain. Okay, you have outer, inner brain, different layers, you have different type of neurons. So this trajectory probably not the time, but it's the anatomic structure of the sample you collected from the cell, from the mouse brain. Okay, so that's all I want to share with you about trajectory analysis. Any questions at this moment?
So really the kind of analysis, we have to make sure the cell, in addition to time, and the factors, the other things to be similar. Otherwise, maybe it's not in time.
That's right. You have other factors which, if you don't know what other factors will impact the gene expression profile for every cells, then you create a trajectory may reflect the change of another factors. That's right. So if you believe time is a major component, just like here, I don't believe time is a major factor to contribute the difference between neurons. I believe is anatomic structure of different layers of brain. So I will say, okay, this trajectory is not pseudotime, just pseudo topology of the cell I collected from the brain.
Do you think it's better to, first reanalyze the neural cells and maybe we have different subpopulations?
Subpopulation.
And
Yes.
you think the different subpopulations to the
Yeah, maybe that's a good idea. Yes. Yeah. Okay, according to our plan, we are going to discuss a separated topic. It's a single cell immunorepertoire profiling analysis. Anyone have any idea what it is? Okay, no idea. Okay. Okay, so immune cells. Okay, they're macrophages, but here we are talking about the T cell and the B cell. Okay, the most abundant immune cells. So on the surface of T cell and the B cell, there are receptors. These receptors define the diversity or heterogeneity of the cell, B cell, T cell population. Every T cell may differ from another T cell because of the different type of receptor combinations. In this receptor, there are subdomains. Okay. Then the combinations of those subdomains change the specificity of this receptor in terms of the binding strength to different antibodies or antigens, not antibodies. Okay, so we want to understand what subtype of those receptors are on the individual cells of T cell and B cell. And then we want to know the frequency of different subtype of those receptor types. We want to know the dynamic if you have a challenge, the infection of the organism before and after infection. Different receptor type may change their frequency. The most important one may increase their frequency of that type. So we want to understand this at a single cell level. At the same time, we want to know within the cell, the gene expression profile. That's a regular single cell RNA-seq. Okay, so we want to know regular single cell RNA-seq for T cell, B cell. At the same time, we want to know their surface receptor, antibody receptor types. Okay, those types are important. They're use that information to communicate with macrophage dendritic cells. But those cells will process information from this antigens, so it can be bacteria, can be virus, whatever, present this information to the T cell receptor, and then to allow different kind of T cell to expand their colony in order to respond to this immune challenge. Well, as I said, this receptor contains multiple subdomains, and these genes was created by either VDJ, the subdomains, and each of VDJ have a cluster of genes to encode their peptides. So you may use V1 plus D2 plus J3 to create this 1, 2, 3 combinations, or you can use V2, D3, J4 to create 3 or 4 different combinations of VDJ. And in that combinations, you give large amount of combination possibilities to to allow this T cell to present this subtype of receptors. Okay. So depending on the immune response or conditions, T cells with particular combinations on their surface protein will get expanded by the clonal expansion. So you just produce this type of the T cell a lot in particular conditions. People want to know, for example, what subtypes in the patient fight with the cancer. Different subtypes will respond for different subtype of the cancer. So that's important information. Okay, so those are the VDJ, they call the segments, okay, where they are located. So subcluster of those genes for VDJ in chromosome 14, on chromosome 7, so different arms, their combinations, their numbers, okay, there are 70 different type of V and 61 different type of J in on the chromosome 14. Okay, those combinations. So once you have, okay, so this is the genome browser view for this T cell receptor, this TRBV. Okay, so you see this subtype of cells, genes. So it's form a whole cluster of many different small genes. So this is in chromosome 14. This is in chromosome 7. So you found the location of those genes. So this shows you the combinations of T cell receptor gene rearrangement. So you start from all this gene, but for each cell of T cell, they're only selected sub segment will express. So this two will express and combine and give you the topology, the combination of this T cell receptor for this cell. Another cell may change this rearrangement. Okay, so large amount of this VDG recombination create a large number of possible combinations, large enough. And also, T cell receptor have two subdomains. So if you create the combinations for the alpha domain, you still have a beta domain to even expand your residual this possible pairs to a large number of the 10 to the 21 possibilities. So from this ocean of these combinations, we have highly selected T cell subtypes under different conditions. Okay. So people want to know what kind of combinations in there. So Telix Genomics recently just published these kits, and so to allow you to characterize single cell T cell or B cell receptor, so amplification key, to allow you to get the information for each individual cell, what their alpha beta chain combinations and what VDG types. And then for the B cell, and you have a light chain, heavy chain, there's immunoglobulin, different subtypes combinations. Okay. So how this is possible? As we know, so this is a system for single cell barcoding. Remember, at the same time, you still have single cell RNA from the T cell, B cell. So we understand this, their barcoding system, okay, their UMI barcode, their Telix Genomics barcode to indicate on the gel bead, and you know which cell the transcript is from. Okay, so these are the same, okay, we discussed. So we don't have to go through all this. Still, this is from a class 1, right? Lecture 1, we know what's the principle of this. All the cells, okay, in this case, majority of the cell will be T cell, B cell, because you will try to work on those cells mostly informative. You try to do this. Okay, this is a major step. So everything what I'm talking about so far, the process is the same. So you have dissociate the immune cells and barcode, and use the droplet, all the GLB covered by genomic barcode, so you know every cell. So once you prepared this, it's ready. In this case, the same sample will be separated into 2 tubes. Okay, one just for regular single cell RNA. Well, another will go this VDG, surface protein sequence. I believe in this step, all the cells have been licensed, all the cell has already broken, all the mRNA has come out. All the mRNA supposed have been labeled with their barcode. Now we separate this sample into 2. Okay, the cell have already been broken, and run the PCR. Sorry, run PCR on your right side. Run PCR for the whole gene. Well, on your left side, only run the PCR for T cell receptor genes. Or should I illustrate this? Yeah. So those are the cells, and then it's already licensed, open up. So you have all the mRNAs here. Those mRNAs were labeled, for example, this is a green, this is red. This is from a 10x genomic barcode. So you know, okay, this is from red, this is from red, and this is from green. So this could be from the same gene. So this gene 1 here, gene 1 red. And then this could be, and then here we separate them. So this is a regular single cell RNA. So this side will be T cell or B cell receptor. Okay. Okay, so this is a regular gene. It will get amplified here. But this gene could be the T cell receptor gene. The specific, this timer will get the sequence for this. And also, you know, this receptor is from the same cell. So later you will know, okay, for this green cell, you will know its receptor expression. And also you know its mRNA expression because you have same label here and you separate them. The green one still here, green one here. Okay. Okay, so this figure to show this T cell or B cell receptor sequencing. You see in the middle, you have this VDJ fragment here. The primer was designed specifically to amplify those regions, not just amplified all other regions. So after you sequence this region, you can reconstruct and say, okay, for this cell, you use V1, D2, J3. For another cell, you use V2, G1, D something other. Okay. Well, this picture is on your right side here. It's regular, amplified order, poly A tail enriched. So all the other genes sequenced here. Okay, so this picture is put them side by side. Okay, on the top is this side. On the bottom, this side. So you see the different. So here you just read other non-receptor genes. Well, on the top, you get information about this VDJ combinations. So the cost of the experiment is doubled. So basically, you sequence this, sequence that, only share the copy in the cloud. Largely, you have to maybe not double, but you increase this part. Only share the part in the middle. Then you put your row sequence in the cell ranger. This cell ranger is a version which particularly designed for the VDJ analysis. It will read out the structure, the type, subtypes of VDJ combinations. Those are the pipeline and some of this output specifically for VDJ analysis. So you run regular RNA-seq pipeline here, and at the same time, because you have VDJ receptor sequence, you run that side by side. And at the end, you merge them because you have barcode shared. The three cell, you know, you meshed, you join those two results at the same time. For the VDJ part, for the RNA-seq part, you still get this UMI matrix, the cell, and all the genes expression. But over the VDJ part, you get something like this. This is just one of the spreadsheet you get. So contigs means different unique combinations of VDJ. Okay, in your sample, you probably have 2,000 of the VDJ unique combinations. And then you list them and see, okay, how many cells has the VDJ V1, V2, J3, and have a frequency of this so-called contig. you can go back and see, okay, for each combination of this contig, what is the sequence? Okay, the rate of the sequence, you can go back to the colony clone type 1, they found this. For clone type 1, they have alpha chain, you have this part, the green part, and the blue part, that's VDJ. And you can map this back to the genome. It's V section 1, D section 2, J section 3. Well, the beta chain have different combinations. Okay, so you have a sequence level of the information because you specifically amplify TCL receptor fragments here. Then you can have regular t-SNE plot for your T cell. In this case, I don't know it's T cell. Yes, a T cell. So because you have this information, you get a T cell t-SNE plot. Well, you also have the receptor combinations. And here I can color code all the T cell in this global clusters contain the same T cell receptor combinations. Okay, we have many different possibilities, but for the sample you have all the T cells share the same VDJ combinations. What that means is all the T cells about this are the same colony created by reproduced or regenerated cell or same type. So this is a kind of response for the challenge. So depends on what the experiment, how you get the sample, so this is information. If you have unchallenged condition, you may have each T cell have its own random combinations. Okay, so they also provide the frequency. This is called bar plot. Yeah. So you see the different colony type and the frequency. So just one is huge in the middle, and the second and the third and have different frequency of the T cells. Okay. That's all. Any questions? Do you think this will be very useful if you study T cell, B cell, and stuff? Yeah.
From the regular single cell sequences.
Yeah.
We also can get some information, gene expression information on this.
Yeah, could, yeah, by chance you should, yeah, but it's not specifically the whole length of the, so you don't have information on VDG subtypes. You may find, okay, V expression level is a 3. You may have some of that information, but not a complete. That's why you want to do a specific amplification from the same sample. So this is more general profiling all the other genes. Okay, good. If you don't have questions, we will meet on Thursday. The video will be available soon if you